Good morning. Good morning, ma'am. Okay, the module that we are going to see today is biotechnology, principles and also applications. So what is biotechnology? What is biotechnology? It is a use of either live microorganism or you use their cellular products or the enzymes that they produce in that you use in, and based on that you develop a process and then you try to get a product which is useful for the mankind. But that you call them as your biotechnology. Actually biotechnology field is not new for us. We were doing it, but we are doing it in a traditional way. So we can study the biotechnology in two phases. One is your conventional biotechnology, the other one is your modern biotechnology. So in conventional biotechnology, if you could see in preparation of curd, wine, in a conventional manner, in a small scale, in our place, that you call them as your conventional biotechnology. So in today's class, what we are going to study is about your modern biotechnology is what that we are going to study. So in modern biotechnology, if you could see what are the two major phases or two major steps which are involved? Yes, good. It is genetic engineering and then genetic engineering, I can even write it as development of rDNA, that is recombinant DNA. And then followed by that, the second process is called as bioprocess. What is bioprocess? My gene of interest I am selecting over here. That is your rDNA or your recombinant DNA. Then followed by that I have to produce them at a larger scale. So that step you call them as your bioprocess. Got it now? So what are the steps that are involved in rDNA technology? Can you tell one by one? Okay, the first step is your isolation of desired gene. Okay, that is the first step. Then cutting off what? Cutting off DNA or the desired gene? Okay, this is my DNA of interest. My desired gene is here. Okay. I have got my desired gene. Step 1 is over. What is the step 2? So now my gene of interest is here. What is? I have to insert it into a host or vector? Vector. I have to insert it into a vector. So this is my vector. So to insert it first, do I need to cut it or not? Yes. So how do I cut my vector? How do I cut my vector? Huh? By using your restriction endonucleases. So I made a cut. I have cut it using my restriction endonucleases enzyme, and then I will add my gene of interest. So my second step is cutting off my vector DNA with, I'm using short forms, right? So with restriction enzyme. That is your second step. Now what is the third step? You have cut, you have inserted. How you have inserted? How you have inserted? With the help of an enzyme called as DNA ligase. With the help of the enzyme DNA ligase. Then what is the next step? Then what I need to do? Now what do I call this as? Earlier it was called as vector. Now after inserting my gene of interest, what do we call this as? Cloning vector. Okay. Then what is the right word actually? Is it cloning vector or recombinant or transformance? This is your recombinant. Okay, and this we call them as your transformants because it has got your DNA over here. Understood now? Now what is the next step? Now I have to insert my vector into a host. Got it? And after that, next step, up to this step, whatever you do is your genetic engineering or your rDNA technology. Got it now? Isolating a DNA, selecting a vector, inserting your DNA into the vector, and then ligating it, and then inserting it into your host organism, and then selecting your host also, the host which has your recombinant DNA. Up to this step you call them as your genetic engineering or your recombinant DNA. So now after you do it, what purpose we have done this whole process? I'm going to get a product which is useful for the mankind. So I have to produce it in a large scale. So that comes here, there comes your bioprocess. And there we will be studying about your, or you have studied about your fermenter. Correct? Large scale fermenter. Two types of fermenter you would have studied. Understood now our overview, what it is and that. So little bit wherever it is required in, I will brush it up and we will see it as a preamble, and then we will start solving the questions. How to answer it we will see. Understood up to this now? Yes, our overview. Any doubt? No. So now what are the tools that are required over here to do my rDNA? First is I used a restriction enzyme. Then I used a vector. Then I used a ligase. Then I used a, what did I use? I used a host. Okay, so about little bit in detail we can study. Okay, what are restriction enzymes? How many types of? Okay, molecular scissors they are called as. Okay, then two types or three types? Two types or three types? Okay, fine. You are telling the major category. Two types. That is your exonucleases and endonucleases. But my question was, already I am talking about endonucleases only, no? Got it? So how many type of restriction endonucleases are there? Three types. Okay, type 1, type 2 and type 3. Okay, which one are we using it? Which type we are using here? For this process, 1, 2 or 3? 2. Please mark it. We use restriction enzyme 2. Okay, because that is what which cuts exactly at your, which side? Which side it is cutting it? Recognition side. Got it? Whereas type 1 will cut far away from the restriction side, whereas type 3 will cut a short distance away from the restriction side, whereas your type 2 is the one which will cut exactly at your restriction side. Got it now? Which is the first developed restriction endonucleases? 2 or 3 or 1? 2. It is your. Okay, but in whole of your book, which is a common restriction and endonucleases that you are coming across? Eco R1 is what that you are coming across with. Okay, you know how to name them? How do we need to name them? Okay, first is the genus ka first letter. Then species, that is your Escherichia coli. Then R stands for your strain. Then first stands for first developed one according to the order. Actually, it is from Escherichia coli RY13. That is a strain. Please take down. It is E. coli RY13 is a strain from which you get this Eco R1. But first developed is your hind 2. Got it? Don't get confused. Okay. So what type of hens, when you use a restriction endonucleases, they will develop? What type of hens they will develop? Sticky hens or blunt hens? Sticky hens. Sticky hens. Got it now? Why we want a sticky hens? So that it is easy for them to go and ligate it. Got it now? So after you cut them with the restriction enzyme, there will be a small sequence which is hanging in right. What is it called as? After you cut, you will have a small sequence which is hanging on either side, right? So suppose if you have G, A, T, T, C. Okay, here also you have your G, A, T, T, G. It's going to cut over here. Okay. It is called as your overhanging ends. This is called as your overhanging ends and we will call them. Got it now? Understood now? Got it? Then following that, what are the other things that you need to know over here? Okay, fine. Next comes your vector. Next comes your vector. Okay, which is the first developed vector? Which is the first developed vector? Okay, what are the different types of vectors that are available first? Plasmid, then phage, that is a virus, a bacteriophage you can use it. Then you can use a plasmid, you can also use a phagemid. Are we studying all this in this lesson? No. Which is the most common one that we are studying? Plasmid. Okay, I think you know what is plasmid, right? What is plasmid? They are circular, extra chromosomal, self-replicative DNA, double standard DNA, which is present in a cell. Got it now? Which is present in a cell. Generally bacteria. Then what is phage? What is phage? If I say it as bacteriophage, you will understand. It is a virus which is infecting a bacteria. Next is your cosmid. That is your lambda phage ka cos site. Okay. Together, that is what which is being used over here. Whereas here, if you could see, we will use a phage plus a plasmid ka combination will be used. Here, cos site of lambda phage plus plasmid you will use. The main idea of using a cosmid and a phagemid is if your DNA of interest is of larger size, then you can go for this method. Understood now? Then comes your, which is the most common vector that you are studying? Plasmid, sorry, PBR322. Got it now? So what is it stands for PBR322? What does PBR322 stands for? Plasmid, based on the scientist name Bolivar and Rodexco, and this is your 322 is a lab name in which it has been developed in. So it is basically a scientist name based on which it has been given in. What are the two selectable markers that you have in the plasmid? Two selectable markers that you have in the plasmid. What is selectable marker? What are the things that a plasmid should have? It should have an origin to start with the replication. It should have a rope or rope region for increasing the copy number to produce them in a larger number. Your interest DNA. Then you have your, what is that? Selectable markers. Selectable markers are your antibiotics. What are the two antibiotic markers you have in PBR322? Ampicillin and tetracycline. You should know that. Got it now? And whenever you select a plasmid, how many restriction site it should have? It should have only one. So whenever you select, it should have one. If it has multiple, don't select it. Got it? That is an idea behind that. Then following that, you have your next enzyme is your ligases. Okay. So during the process of ligation, to add your to ligate them. So usually ligase prefers, it creates phosphodiester bond between 5 carbon site of one nucleotide with that of 5 carbon site of another next nucleoside. So here you will have an hydroxyl group, here it will have a phosphate group, and it will create a phosphodiester bond using that. So remember that ligase creates phosphodiester bond. It usually forms a bond between the 5' end of both your nucleotides, 5 carbon points. Okay. So one will have a phosphate group, to another end you have an hydroxyl group, to that it will link in and then it will form in. Got it now? Understood? Next is your host. Which are the two common organisms which will be used as a host? Animals. Generally, if they ask, options only. If it is bacteria, it is coli. Other than that. Fast, you should answer now. Next is your yeast. They use, I have given the hint already, yeast. So you should name the yeast. Commonly studied yeast. Saccharomyces cerevisiae. That is what you are studying. So only that yeast is available. Got it? So next is your Saccharomyces cerevisiae. Got it? Now these are the two commonly used host. Okay. How you will insert your DNA into the host? How you will insert your Can I erase this, or at least half of it I will erase? How you will insert them? Gene gun. Okay. You say I will write gene gun. Next step. Micro injection. Right. Then remember vectorless method. If the question is vectorless method, you can go for both of this. Gene gun usually we use it for which cell? Plant cell. Whereas micro injection is used for animal cell. If my host is a bacteria, what I should do then? If my host is a bacteria, what I should do? Huh? Okay, we make the host competent. Do you remember that? So what I have already given the hint, competent. Which method we will use for this now? Calcium chloride. Treat your E. coli with calcium chloride. What will happen? It will increase the cell wall permeability. It will create larger pores. Then what you do is that you add your RDNA, that plasmid which has that RDNA, that is your vector, plus your host bacteria, both subject it to first calcium chloride. Then keep them onto your heat shock condition, that is 42 degrees Celsius. Then keep them on the ice. This will enable them to take their, what is that, vector into the bacteria. This is the method that we follow. Understood now? Got it? Yes? An overview of this lesson? Any doubt? Okay, now whenever we are cutting our host DNA, that means the gene of my interest, I have to take only my gene of interest, right? After cutting it with the restriction enzyme. So what is the technique that I will follow for that? Please don't talk. What is the method that I will follow? Jelly electrophoresis. Okay. Jelly electrophoresis that you will follow in. So what is the basic principle on which the gel electrophoresis works in? Based on your size. The molecule ka size. Got it? Generally remember, first point, DNA is negatively charged. So it will move towards your positive charge anode. So whenever you load them into the well which is negatively charged. They are negatively charged. Towards the cathode you load them. So they will start to move. Based on their molecular weight, that is your size, you are separating them. Usually the less molecular weight will be moving farther. One. You are able to get it. And how you will visualize them? You will visualize them based on your ethidium bromide and observe it under your UV radiation. They will appear it as your orange in color, orange band by which you can pick them. So once your gene of interest is there, then what we will do is we will do a process called as your elution also. That is, elute them, isolate it and then insert it. This is one step. Got it now? And the last process is your bioprocess, for which we use fermenter. Generally we use fermenters because I can produce my desired product in a larger scale. One, and optimum conditions it will produce in. What are the two type of fermenters that you are studying in? Stirred tank fermenter and also your sparged fermenter. Got it now? So optimum temperature, nutrients, pH, everything is controlled over. You are able to get it. In case of sparger, we will flush the oxygen from down so that there is a uniform distribution of oxygen. Got it now? This is your first lesson. That is your biotechnology ka principle. Your second lesson starts or it talks about your application. What are the different application of biotechnology or where all it has been used in? In which all field we have used them? My question is in which field? Please don't talk. Agriculture, then? Okay, research where? What till now what you have done itself is a research. Okay, because you are finding out a gene of interest. That itself you are researching and finding out, right? So in which all field you can use them? Agriculture, then medicine, then Bacillus thuringiensis, right? So how does this principle works in? Crygene. So crygene is coded by which gene? It produces which protein? Cry proteins. What are the different types of cry proteins that you have in? What are the different types of cry proteins that you have in? Cry1AC, then Cry2AC, and then AB also. And you should know that for corn borer we use Cry1AB, whereas other two are used for your bollworms and your armyworms. Generally, insects which belong to the category of Coleoptera, Diptera, they can handle in. Okay, they can kill in. Got it now? And it works on a alkaline pH. Got it? In the insect gut, it acts on him. Got it now? Okay, one more thing. How do they introduce this gene? Which is a vector that is used to introduce this gene, BT cry gene into the BT cotton? What is a vector for the plants that you will use? In plants, which one is a vector? In plants, directly I want to insert my gene of interest into the plant, which is a vector. Can I use plasmid? Can I use plasmid? Just yes or no. Can I or not? No, you cannot use it. Then, what is the next option? Yes, that is right. You can use TA plasmid, but which organism has that? Agrobacterium tumefaciens. So, please remember. Got it now? So, in case of your plants, if you want to directly inject in, you use TA plasmid and the bacteria which harbors is your Agrobacterium tumefaciens. Okay, that you can use them. Then next, other than that BT cotton, then what are other products that we studied in agriculture? Pest resistant. Which is a pest resistant that we studied in? Root knot nematode that we studied in. What is the mechanism that they used in? RNA interference, that is gene silencing method, that is double standard RNA will be produced in, okay, by using the complementary sequence of DNA, and whenever they have this, then the translation of the double standard RNA cannot happen in and silencing of the gene. So the protein which is essential for the nematode cannot grow, so the nematode will die. So the technique is your RNA interference. In that double standard RNA will be produced in. Messenger RNA translation cannot take place. So as a result, protein required for the parasite cannot be produced in. Got it now? So that's a technique that is involved over there. Other than that, in agriculture, and even including your biofortification. Okay, BT tomato, right? Flavor saver. Flavor saver. What does it mean, flavor saver? Flavor saver tomatoes. What does it mean? They delay the process of ripening in tomato by introducing a gene called as your polygalactourinase. Usually tomato get ripened at a faster, so commercial value won't be there. They want to delay the process so that the farmer can hold it and sell it for a longer duration. It is for your transgenic tomatoes that we will produce it. Next, rice, golden rice. What is golden rice? Vitamin A rich fortified rice. It will be yellow in color, so it is called as your golden rice center we will call. But do we introduce vitamin A ka gene directly? No. Which gene are we introducing over here? beta carotene, which is a precursor for your vitamin A. So when this gene is there, beta carotene will be produced in, and from beta carotene vitamin A will be produced in. Got it? That's how it happens. It understood now? These are some of the bioquantifications which happens in your agricultural field. Then in medicinal field, what we have studied? Gene therapy. Okay, gene therapy I will write in. Therapeutics I will write. In medicinal field, again insulin. Got it now? So the insulin which is produced by recombinant method, what is the name of it? What is it called as? Humulin. Humulin. It is called as humulin. Okay, earlier from the pancreas of pigs, cattle, they were preparing it. Got it? It has lot of side effects. So they have shifted for the recombinant DNA technology. But the major problem with the insulin development is that how many chains it has got? How many chains does the insulin will have? Two chains. Actually, it has got A, B and C, 3 chains. But the third chain is not required. So naturally in our body when it is produced, the C chain will be removed off during your maturation process. So that they were not able to achieve in a recombinant DNA technology. So what they did is that they used E. coli, they inserted for A peptide gene, another E. coli host cell, plasmid B. So this will produce only A chain, this will produce only B chain. Then they combined both of them using the disulfide bond. That's how they did. Which is a company which is responsible for it first, which developed? American based company, Eli Lilly. Okay, they are the one who first developed this. Got it now? Then other than that, we have even used to treat, it is used in gene therapy. To treat, in gene therapy where we are using them? To treat which disease or which condition? ADA deficiency. What is ADA deficiency? Okay. There is a mutation in the gene which codes for ADA. Got it? So as a result, the person will be suffering from SCID. Please don't get confused. The deficiency is ADA gene ka deficiency. But the condition what the patient will suffer is SCID, that is severe combined immunodeficiency disease. There is a lack in the functional lymphocyte production, especially your T lymphocytes cannot be produced in. What is the use of T lymphocytes? What is the use of T lymphocytes? Yes, study by over no human. What is that? Human disease. How many different types of T cells do we have? T helper cells and T cytotoxic cells. T helper cells will even help the B cells also. So if T cells are not produced, then B cell will not do their function. So you will not have humoral mediated immunity or cell mediated immunity over here. Total immune system will be affected. Got it now? So how you can treat this condition? That is why it is called severe combined. That means combined of both cellular and humoral immune deficiency. That's a severe combined. Now tell me, how do they treat this condition? How you can treat this condition? Generally by bone marrow transplantation they do it. Got it now? But here in this technique what we are doing, in recombinant technique, we are isolating some of the lymphocytes from the patient, inserting the gene which codes for ADA and putting it back. So the person will start to produce lymphocytes. But lymphocytes are short-lived. So again and again, every time you have to keep on doing this. Okay, but he can be there. But if you do this step at the embryonic stage, he can survive for a longer time. Got it now? So that is one. Next, following that, next, in diagnosis, PCR. What is PCR? Reaction. Yes, what is ELISA? Enzyme assay. That can also be used. So here, even a small amount of the pathogen, if it is there, before the symptom could develop it, you can amplify it and it can be identified. What are the 3 steps which are involved in PCR? Denaturation, annealing and exhalation. Yes. Then what is the enzyme which is used in the PCR? Thermus aquaticus. That is your Taq polymerase enzyme which has been isolated from Thermus aquaticus. We use it. Got it now? Understood? You are able to get it? So then, what is that? ELISA. In ELISA, it is based between antigen and antibody interaction. Okay, whenever any infection that sets in, immediately there will be an antigenic body, a foreign substance. That foreign substance can be detected over here. It is based on your antigen antibody reaction. So here you can detect HIV, HPV, all pathogens can be detected in. In case of PCR, even you can detect a gene which can cause cancer also. That gene can be amplified and they can be detected from the patient. So all this that you can do it. Next is your last one is your in therapeutics. Interferon production, human growth hormone production. Got it? And then what is that? TPA, that is your tissue plasmogen activator production. These are all some of the therapeutic substances that are being produced and which can be used. Got it? Now you are able to get it? Yes. So you have a lot of scope in case of your biotechnology. And how many recombinant proteins till now they have developed? Till now they have, how many of them have been developed? 8. 8 is marketed. 8 is marketed. But how many is there? Almost 34 recombinant proteins in the pipeline, but 8 is been in the marketed stage. Got it now? Then last part is whenever we are inserting a gene into an individual, into a bacteria, automatically we are doing unethically. We are exploiting them. So there is a control which is required for it. For that we have a body called as your GEAC. What is GEAC? Genetic Ethical Approval Committee. What is the function of this committee? This committee will check whether the gene which has inserted is going to cause a harmful effect on the host, any side effect that it is going to causing, or it is going to affect the environment or the ecosystem, everything, and at what amount they are exploiting even the resources, everything will be checked over here. Okay. So in pertaining to that, we are also studying biopiracy. What is biopiracy? Taking our resources from a country without that knowledge or without that people and exploiting it for my benefit in another country. Most of the multinational companies do it. When they go in, they take their product or they take their plants and animals, they come, but we don't take the permission or give the monetary benefits also for them. We don't do it. So that regulation is called as your biopiracy. Okay, so if you could see in, there are 3 patents which are reversed back. One is your basmati rice, which is a US patent. Usually there are 2 patents in the market. One is European, other one is your American, USA. Okay, so in both these cases, turmeric for wound healing property, US patent they have got. Similarly for basmati rice also they have got, but it has been later cancelled. Got it? In case of neem, it is a European, what is that? Patent that has got in to say that it has got fungicidal activity. Later we approved it and then that is also been cancelled. Got it now? This is the overall gist of your biotechnology and its applications that are being involved. Understood now? Yes. Shall we start solving the questions one by one? Yes, ma'am. Yes. Here is an example. Okay. Second generation vaccines are prepared by recombinant DNA technology. Which out of the following are examples? Tell now. Option D. Both D. Okay, fine. First recombinant vaccine is hepatitis B vaccine. If they ask what type, it is subunit vaccine. Got it now? Next, it is your herpes virus. Correct. SARS polio vaccine is a killed vaccine. It's not a recombinant vaccine, right? If they ask the third one, what you will write? 2 is given over here, A, B, right. Third one, take down. It is even pneumonia vaccine if they give. Third option. Okay, pneumonia vaccine is also a second generation vaccine, recombinant vaccine. It's not given, but note it down when you are doing it so that in the options we don't know. They may give that also. Now next, read the following statement and select the correct ones. Select now. Electrophoresis is a technique used for the separation of substance of different ionic properties. Right or wrong? Right. Next, plasmids are extra chromosomal, self-replicating, circular, double standard, and naturally found in bacteria and also in some yeast. Right or wrong? Correct. Then third one, it is not advisable to use an exonucleases enzyme while what is the keyword which is wrong over there? Endonucleases. Yes. The last one is eco R1, Roman letter 1 indicates that it is the first type of enzyme isolated from eco stain RY3. Yes or no? Yes. So what is the option now? C. No. D. C. Yes. 1, 2 and 4. Yes. C it is. Right. A specific recognition sequence identified by endonucleases to cut at a specific point within the DNA. What is it? C. That is your palindrome sequence. How do you read a palindrome sequence? In both the stands, either reading them from the forward to the backward and from the backward to the forward direction, it should give the same sequence. Not in one. In both the stands, you should consider it. Got it now? So now mark the scientist here. Carry-Mullis. Remember, Carry-Mullis is always for PCR. Got it? He developed the PCR chain reaction and he was given Nobel Prize also for that. Next, Paul Berg, father of genetic engineering. Third one, Stanley Cohen and Herbert Boyer. Okay, what is the specialty of these two scientists? They are, yes, right. They are the one who first developed this rDNA technology. How did they do it? By taking the antibiotic resistant gene from a plasmid of the bacteria Salmonella typhimurium. But where did they insert it? They inserted that gene into plasmid. Don't get confused. Their antibiotic resistant gene they took from Salmonella. But when they constructed the recombinant, the host is E. coli. And check, normally E. coli is not antibiotic resistance. When they introduce that gene, it becomes resistant. That's how they did. Don't confuse between where it is being used in. Next, obviously the last one. You don't need to think only. Even if you don't know also, you can write, right? Yes, he is the one who developed the restriction endonucleases. Next, what is the option now? A. Next, different steps of recombinant DNA technology are given below randomly. Now arrange and tell me. Which is the first step? Isolation of DNA or genes to be cloned, right? Then what is the second step? Insertion. That is what you missed in, right? The second is insertion into the vector. Then the third step? Option C, right. Next, gel electrophoresis is a
technique separation of DNA fragments through the pores of across and under the influence of electric field.
Very good correlations, right? It's right also. It's a technique for separating your DNA fragments through the pores of agarose gel under the influence of electric field. Magnetic field purification. Okay, now again, match. Bioreactor.
Production of large
Quantity of products. Then electrophoresis.
separation of DNA fragments.
PCR amplification. See, this PCR and gel electrophoresis, if you consider, it's a technique. Wherever you want to separate DNA, you can use. Got it? Similarly, PCR also. Don't think PCR is only used in case of your, what is it, gene amplification when you are cutting it. Even in diagnosis, I can use this because I just take the sample from the patient and then if they have a small amount of the virus also, it will get amplified over it. So, wherever you come across with amplification, PCR will be there. Techniques can differ. Next, what is the option then?
B.
Next, B or A? B. Next, the role of DNA ligase in the construction of recombinant DNA molecule is?
Formation of phosphodiester.
Understood, right? Where do they form between? Between your 5' carbon of one nucleotide and to the other phosphate and the hydroxyl group. Next, first discovered restriction endonucleases. D. Hint 2. Next. If a recombinant, 10th question, if a recombinant DNA bearing gene of resistant to antibiotic ampicillin is transferred to a E. coli, the host cell become transformed into ampicillin resistant cells. If such bacteria are transformed into an agar plates containing ampicillin, only transformants will grow. So, what do you call this as? Okay, now there is one more concept. What is insertional inactivation? What is insertional inactivation? If you insert a, what is insertion inactivation? They will ask. Okay. So insertion, in case of insertion inactivation, here when you insert the gene into the selectable, into one of your site, then your antibiotic becomes lost, resistance is lost. You are inactivating the gene for the antibiotic resistance, right? So similarly, there is an enzyme called as beta galactosidase. This is another method that you can use. The gene which codes for this enzyme is lac gene. Got it now? So this enzyme acts on X-gal. X-gal is a substrate. An enzyme will act on a substrate. Substrate is X-gal, commonly called as BCIG, bromochloroindoyl galactosidyl. Okay, but you don't need to remember. Only short form if they give you remember. X-gal is what that they will ask it. You are able to get it. So whenever the enzyme, if the gene is there, enzyme is produced. The gene is there, enzyme is produced. Yes or no? Yes. And the enzyme will act on X-gal. Then X-gal will be converted into, or it splits and produces a insoluble blue colored compound. When it splits, it produces a blue colored compound. Got it? Suppose if there is no gene, will the enzyme be produced? No. Then automatically it will not act on X-gal. So your colonies will be white. So which one you will select? White or blue colonies? Blue colonies. Okay, so this is called as insertion inactivation. Okay, so now my question is, which one you will select? Antibiotic or beta galactosidase? Why not antibiotics? If you have a vector or you have a method to use this and even the other one, antibiotic marker, which one you will prefer? They will ask this. Beta galactosidase only you will prefer. Why? Why? Because in case of your antibiotic, the procedure is cumbersome. You are inactivating them, preparing the plate, again selecting them. The procedure is cumbersome. That's all. Got it? So we will go for beta galactosidase. Next, which of the following should be chosen for best yield of one has to produce recombinant protein on a larger scale? Which one you will select now? Laboratory flask of large capacity, still tank fermenter without inlet and outlet, a continuous culture system, or any of these. Any one you can go in because you want a larger production, that is best yield only you want. So you want a larger capacity also, continuously production also, and a still tank without, that is your static method. Okay, any of these, D is an answer. Next, read the following statement and select the correct answer. Same kind of sticky ends are produced when a DNA has been cut by different restriction enzyme. Correct or wrong? Same kind of sticky ends, that means same cut. Only one kind of sticky ends are produced when a DNA is cut by different restriction enzyme. Different restriction enzyme, different site. But always sticky end. Always sticky end. But my cut need not be at that point only, because all vector will not have the same sequence. So that is wrong. Next, exonucleases makes cut at specific position within the DNA. No. The wrong is exonucleases. Next, HINE2 was the first restriction endonucleases. Right. Then next is a bacteriophage has an ability to replicate within the bacterial cell. Yes. partition site within a vector facilitates a gene cloning? No. If you select a multiple cut, then you will get a multiple pieces of the vector. How you will join them? What is the need? Got it? So never we will go for, not even more than one we will select. More than one, don't select. Okay, next. So the option is C.